later. Better late than never. Um, as much as I like to think I'm a scientist, we found it imperative to assemble an expert scientific advisory board. The board helps us outline and modify our research roadmap, target potential projects, and most importantly, review our projects to make sure they are feasible and structured so they are inclined to succeed. No one on our board is funding, allowing them to be completely unbiased. And so I just wanted to introduce our scientific advisory board to all of you. We have Rachel Bailey, who is a gene therapy expert, Alessandra Bellino, who is a CMT4 type 4 expert, Laura Feltry, who is a animal model expert. And then we have Dr. Caroline Martinez, who's Hunter's developmental pediatrician and works closely with a lot of other families with children with rare genetic diseases to help them build their roadmaps. We have David Parison, who is another CMT expert in Italy, who actually has been very helpful in helping us identify other CMT4B3 patients throughout the world. And then we have John Starin, who's a molecular biologist who is very involved with the CMTA. He is their scientific chair, so he's aware of all of the scientific research that's being done on CMT throughout the world. And so it's really helpful to making sure we're not duplicating any research and to keep our research as streamlined as possible. So because of all of you, we have initiated 11 different research projects, 8 of which we are solely funding. So I just want to go through them all with you really quickly so you can see where your donations are going. So in just an alphabetical order, we have Dr. Robert Burgess at Jackson Laboratory, and he is examining SBF1 mutant mice for CMT4B3 relevant phenotypes. So basically, he's our mouse guy. He has our mice up in Maine, and he's trying to see if he can see how they look sick so we can test some drugs on them. Then we have Dr. Matt Butchback at Nemours Alfred I. DuPont Children's Hospital, and he is taking patient samples and turning them into different cellular models. And he's also looking at our mouse model. And you can see the grant amount we have granted each of these people and the length of the project up on the left. We have Dr. Jim Dowling at 6 Kids. He is making our zebrafish model for us. And once that is up and running and they can see the phenotype saying how sick, how this animal shows it's sick versus a normal zebrafish, we can start screening drugs on those as well. We have Dr. Jefferson Doyle at Johns Hopkins University, and this is Hunter's ophthalmologist who we met at Johns Hopkins, and he was kind enough when we met him to say, hey, I do a lot of research on rare genetic eye diseases, and he has some of our mice down there now, and he's looking at their eyes and trying to see if there's anything wrong with them. And we're in Boston right now. We're coming back Friday, and then Sunday we're off to Baltimore to go see him. So we're very busy, but he's so nice. And then we have Dr. Scott Harper at Nationwide Children's Hospital, who you will be hearing from tonight. He is doing one of our gene therapy projects. We have Professor Henry Holden at UCL Queen Square in London, and we are so lucky to have him on our team because he actually has seen other CMT4B3 patients, and he is examining Hunter samples and all the other patients that we can include in our natural history study. And a natural history study is really, really important for when we develop a treatment for FDA approval. So we're glad we have that going. We have Dr. Laura Lambert at University of Alabama. This is actually one of the programs that we initiated that we are not funding, and she's in the process of developing a rat model for us. Dr. Jennifer Moore is at Sampled, and she has made our stem cells for us. And she also is where we host our own little personal biobank. Dr. Mario Saporta at University of Miami. And he is taking those stem cells that we developed and turning them into motor neurons and these spinal spheroids. And he helped with other rare types of CMT develop a treatment by using those type of models to see if they could test certain drugs on them. So we're hopeful that he will do the same for CMT4B3. And then we have Dr. Stefan Zuckner at University of Miami with his mini gene construction and validation. And this is our second gene therapy project that we are funding. And it is really just a very, we're really happy to have Stefan on our team because he is one of the lead geneticists in CMT. And now I just want to make sure that I can see the list of my participants, but I want to make sure that Scott is on this Zoom before I introduce him.
I'm here.
Oh, great. Okay. So, and without further ado, I'd like to introduce Dr. Scott Harper. Those of you who were at our Zoom kickoff last year will remember Scott's amazing gene therapy work. Scott has gone above and beyond even before we funded him to try to solve the problem of how to fix the genetic mutations that cause CMT4B3, and he has been one of our biggest supporters, and he has been working really hard on trying to solve this very complex problem. But I will let him share more of that with you because he is a, I'm sure will say it much more eloquently than me. I just want to make sure I have given him ability to share. Okay. Hi, Scott.
Hi, thank you.
Thanks for coming.
Nice group here. Looks like maybe 30, 40 people.
Yeah, I think we're up to 47 participants, and I know there's a bunch of people that have 2 people or more on their screen, and we're just really happy to have so many people that care about Hunter and helping us raise the funds to develop a treatment for CMT4B3. We know you're our guy, Scott. We know you're going to figure it out.
Well, I hope so. So, you know, it's easier to read the room when you're actually in the room with people. In Zoom, it's a little bit harder. Do we have any scientists in this group? Anybody?
We have a bunch of doctors.
Okay. Okay. All right. Well, I have a couple of slides I want to show just to sort of, so we have one hand up there. So I have a couple slides I want to show, and what I really want to do is just give you sort of an overview of what we're trying to accomplish. So you may have heard, you know, talking to Iris that Hunter has a gene mutation in a gene called SBF1, and so you know he doesn't have a functional form of this gene. So you know we have for most of us. For most genes, we have 2 copies of a gene. So we have one from our mom, one from our dad, and sometimes we inherit mutations in both those copies of those genes, and that can give rise to genetic disease. And you know, so what I work on is gene therapy, and what that means is actually why don't I share here. There we go. Can you see my screen?
Yes.
Okay. So gene therapy, hold on here for a second, just went black. Are you seeing my screen now?
Yes.
You're seeing my slides, not the gallery?
Yes.
Because I'm only seeing the gallery. Okay. I might have to minimize this. Okay, there we go. So gene therapy is defined as treating disease with DNA or RNA. So what this really means is, so for somebody who has so in Hunter's case, he's missing functional gene this SBF1 gene, right? So gene therapy is actually a very sort of straightforward concept, which what we want to do is for somebody who's got a gene that's missing, we want to add it back. So that's really the definition of gene therapy, is treating disease with DNA or RNA. And so our goal then is to try to Hunter has been identified to have 2 different mutations in both copies of his SBF1 gene. So he doesn't have a functional SBF1 gene. We want to add back a normal copy to his cells, in particular his neurons mostly, and hope that can correct the disease that he's experiencing. And so this is not, it sounds like science fiction, but it's actually possible. And there's precedence for this now. And I'll tell you, I talked to this group, or maybe some of you remember this, I guess it was last year around this time. And I presented a gene therapy that's already approved by the FDA. I'm gonna show that again for the benefit. I think to see these things twice is helpful, but also there are probably some people on this Zoom call that haven't seen this before. But I do want to sort of set the stage for what we're trying to accomplish here. So how do we add a gene back? So we have a gene that's missing. We want to add it back to cells. How do we do this? Well, I mentioned DNA and RNA. DNA and RNA. So what I'm showing on this slide is we have this represents a cell, and this is the cell membrane. And then here's the nucleus of the cell. This is where the DNA is located. So you can DNA and RNA are charged molecules. They're chemicals basically, and they're negatively charged. And our cells, these membranes that are shown right here, have evolved to keep out DNA and RNA naturally. So they want to keep out foreign DNA and RNA, and they want to keep our DNA inside the cell in the nucleus. So our cells are very resistant to uptake of these molecules. So we can't, it's hard to just add some DNA to a cell and expect it to go in and be able to correct a gene that's missing. So the method that we're using, and the method that most people in gene therapy are using, is to use viruses. So we essentially hijack viruses to use as delivery vehicles to deliver DNA to disease cells. So the virus that we like to use is called an adeno-associated virus, AAV. And so these viruses they're present all over the place. Like you just walking around in the world, we are encountering these viruses, and they don't cause disease in humans. But the one cool thing there's several cool things about them, but one of the cool things about them is that they I have skeletal muscle here listed on this slide. They like to infect skeletal muscle, but they also like to infect neurons. And they also can infect cells in the eye and the liver. And when they do that, this is what happens. So the virus, so this represents a virus, and it's got this DNA inside it, its own viral genome, its own DNA, and it can bind a cell. It gets internalized in the cell, and then it moves to the nucleus of the cell, which is where our DNA is located, and it releases its own DNA. And so it uses the virus essentially injects its own DNA into our cells, and then uses our cells to produce more virus. That's sort of the general life cycle of a virus. Well, for gene therapy, we have figured out a way to hijack this system. And what we can do is we can take out the virus genes. We can replace those with genes that are, what we call therapeutic genes that are missing. So for the example of Hunter, we can insert, what the goal is to insert an SBF1 gene, a functional SBF1 gene, inside this virus, remove all the viral genes, grow the virus up in the lab, and then deliver that back to the cells and use the virus's own machinery to inject a good copy of the SBF1 gene in the neurons of Hunter in this case. So this is the idea. We're using viruses as a delivery system to deliver DNA, good copies of genes, to cells. And it sounds, like I said, it sounds like science fiction, but this is actually feasible, and we can do this. I mentioned that these viruses, they like to infect muscle, they infect heart, liver, brain, so neurons, and then they also infect the eye. And there is precedence for this. So there's a couple of, so these virus systems have been used in clinically approved, FDA approved gene therapies for blindness and for spinal muscular atrophy. And there are a number of other ones in the pipeline, including some work we're doing for some forms of muscular dystrophy. So I just want to show you a little bit about how these work and some of the work that's been done in animal models and in people to demonstrate that these viruses are good delivery systems for gene therapy. And so the same virus system that I'm telling you about, AAV, was used in the first FDA-approved gene therapy product. It's called Luxturna. Oops. It was developed by a company called Spark Therapeutics out of Philadelphia. And it's remarkable. It is literally a cure for blindness in children. And I'll show you some of in the next couple slides, I'll show you some videos on how this works. And I apologize to people who've seen this before, but I think every time I see this, it moves me. And I think it's a really cool example of how we can use gene therapy to treat disease. So in this disease, this blindness disorder, it's called Leber congenital amaurosis. And it arises in a similar way to CMT4B3, where there's two copies of a gene called RPE65 in this case that are defective. So they're not able to produce a normal RPE65 protein. And when that happens, people who have inherited these genes that are missing, they become blind. And so this therapy, Luxturna, was developed to use this virus system to deliver back a good copy of the RPE65 gene. And the way they're doing this then is they're using the same viruses I told you about. They're cloning this gene inside this virus. They're growing the virus in the lab and then injecting it in the eye of these children. The one other thing I want to say, and I'm going to show you the evidence for how this works in the next couple of slides, but the other thing I want to point out about gene therapy is that this, if it works, this is intended to be a one-time treatment. So these viruses, if you inject them into cells that aren't dividing rapidly, like neurons, like muscle, like cells in the eye, they'll just kind of hang around. They get into the nucleus of the donor or the recipient cells, and they just sort of stay there, and they start producing this gene indefinitely. The longest we've ever, that when I say we, I mean our field, the gene therapy field, has looked is 10 years. And so we know that human beings that have injected with these viruses 10 years later are still producing the gene product that's coming out of these. I mean, this is illustrated by Spark's NASDAQ stock ticker. When they went public, their stock ticker is called, it's once. So this is supposed to reflect this idea of this one-time treatment. Okay, so let me show you, I'm going to show you two slides here with videos. And so one of them shows the importance of using animal models to test therapies like this, gene therapies. And so I've got three videos here on this slide, and this is some of the data that was used in a mouse model to demonstrate that you could use a gene therapy approach to replace this gene RPE65 and treat blindness. So in the first video, what you're seeing here is these green images. These are water tanks, and this is shot in the dark. So this is night vision. That's why it's green. And so what this is the edge of a water tank. There's a person here, and this person's holding a mouse. And then down here is a platform. And so the idea here is that you drop the mouse in this tank, this water tank, and the mice obviously want to find, they're going to swim until they find a platform to be safe, right? They don't want to drown. So they're going to navigate this water tank by vision, by sight. And so this is, in the first video, this is a mouse model that has a mutation in this RPE65 gene. And what you'll see,
Young experience RD.
there's the mouse swimming.
Now, trial 4.
And he just can't quite find, he can't see, so he can't find the platform. And this goes on for a while. I'll stop it, but the video, the mouse never finds the platform. Okay, so the second video is the same experiment, but now we have a mouse that has normal vision. So this mouse can see, and you'll see how this animal performs. Okay, so right away it finds the platform. Now this video is the same mouse model that we saw in this first video, but this mouse has now been injected with this virus carrying the missing gene. Now in this experiment, the scientists here have added back the gene to the eye of this mouse. And now you can watch this video.
Page 12, number 2, RD treated, trial 2.
So I think this is pretty cool, right? So this demonstrates that if you've got this mouse model that can't see, you add the gene back using this virus system, and now the mice can see and they can find this platform. And so they did a lot of other experiments to demonstrate that this is functional. And they did enough experiments and enough, so they did experiments to show efficacy, and they did experiments to show that the treatment was safe. And once they established those two different parameters of efficacy and safety, they could go to the FDA and say, okay, we want to test this in human beings. And so that's what the next video shows. One of the clinical trials where this was done, the same vector product, using this virus to deliver this gene to the eye of a little boy who's blind. And so in this experiment, now they injected only one eye, and they left the other eye untreated initially. Okay, so in this case, what they did was they injected this little boy's eye, and then they patched the eye that was injected. And then they asked him to navigate a maze. And so what you'll see in this video is it's just a rudimentary maze in a room, but they have these arrows on the floor, and the boy is supposed to follow the arrows to get through the maze. And so again, he's been treated, but they covered his treated eye, so he's got a patch over his treated eye, and you'll see how he performed.
I'll tell you if you're going the right direction.
At least I can see the black squares.
Okay, stop. Okay. Turn right.
It was hard. This is hard.
So this goes on, this is like that first mouse video, this goes on for quite a long time, and he doesn't make it through the maze, he can't see. So in the next video, I'll show you, all they do is they take the patch off the treated eye, and they cover the untreated eye, and they ask him to navigate the maze again. So this is just a few minutes later, same kid.
Perfect. All right. And you can start whenever you're ready. Wow.
Wow.
Every time I've seen this video probably like 25 or 30 times. I teach this in a class at Ohio State, and every time I watch it, it just gets me. I should think it's amazing. Okay, I'm gonna stop for a second and I just wanted to set the stage for what these virus systems are, the concept of gene therapy, what we're trying to accomplish here, and then how are we trying to use the same sort of idea to treat Hunter. It's a little more complicated because, if you think about this, one of the reasons why, this is remarkable, but one of the reasons why the first gene therapy that was approved and had been made it through this sort of difficult pipeline of safety and efficacy. One of the reasons why that it was treating a blindness disease is because the eye is relatively easy to treat, right? It's a small organ. It's immune privileged. And so we don't need a lot of virus to treat the eye. And there are only a few cells that we have to treat to have some benefit there. Now, diseases that more widely affect a person, for example, muscular dystrophy affects all the muscles. That's 40% of body mass. That's what I work on primarily, is muscular dystrophy. But also in Hunter's case, we want to treat neurons, and probably, we have millions of neurons that need to be treated. And it's a lot trickier to be able to deliver this virus system to hit these neurons, to hit as many neurons as we need. Number one. The other issue is different genes are different sizes. So the RPE65 gene that was used in this blindness study, it's pretty small. So it's small enough to fit inside one of these very small viruses. And so we can do the molecular biology, we can grow the virus pretty easily. And again, I say we, I didn't have anything to do with this. I'm using we as like a representative of the gene therapy field. But to be clear, this is not my work. I don't want to leave the impression I'm taking credit for any of this. But as scientists, we want to be able to employ the same system because it's so powerful. The problem with SBF1, in addition to having to treat a much greater number of cells than what was required for this particular disease, is also that the SBF1 gene is too big to fit inside this virus. And so this becomes sort of a DNA engineering problem now. Now we have to figure out a way to reconstitute this big gene. How do we package this big gene inside one small virus and use that virus to deliver it? It's challenging. And so Iris mentioned that there's another lab, Stefan Zuckner's lab, who's trying to do this by shrinking down the gene. So he's trying to identify important regions that might be that we have to keep, and maybe some regions of the gene that are disposable. So can we trim this down to make a smaller version that will fit inside these viruses? Our approach is different. What we're trying to do is basically cut the gene in half and package the first half of the gene inside one virus and package the second half of the gene inside the other virus, and then deliver both of those viruses to the same cells and hope that they can recombine inside the cell and stitch together the full-length version of that gene. Now, this is challenging, and we knew this was challenging when we first started, when we were first discussing this project. But there is precedence for this. People have done this for other diseases. It just requires some trial and error and some engineering. And okay, so I'll jump back in and I'll just give you the sort of the basic overview of what we need to do here, and what we're trying to do. So let me share again one more time our approach. And this is just on one slide, and I hope this isn't, so please, everybody, please stop me if this becomes too confusing, but I want to make sure I'm communicating this effectively. Can you see my screen?
Yes.
Okay. Is it big enough?
Yeah, it's big.
I can make it. If I do this, then I can't see everybody. I can't see the gallery, so I don't know if anybody's raising their hand or something to interrupt. So please, if I'm not making sense and you want to interrupt me, just speak over me, because I can't see the gallery to see the hand raised thing. Okay, so again, we're trying to package this long gene, this big gene inside the small virus, and we can't do it as one piece, so we have to cut it in two. And there's 3 different ways that this can be done, and I'll start it here at the top: A, B, and C. Now, a lot of this is scientific jargon, so I'll try to not use the jargon and explain what I'm showing here. Okay, so each one of these sort of cartoons, these boxes, represents the DNA genome of a virus. And it's not the virus genome itself, it's the thing that we're engineering. This is the gene. So this is the first half of the SBF1 gene. We call it the 5' half. This is really just the first half. So we've got one virus we've made, and we've done this experiment already. We've made this virus, and then we've made the other half of the virus. So in this first experiment, what we're trying to do is co-deliver these 2 viruses, and we have some overlapping regions. So these lines here represent overlap. So this is the same DNA sequence present in here, in this copy, and in this copy. So we basically maximized the amount of overlap we could and still fit these into the virus. And the hope here was that if we delivered this virus carrying this piece of DNA and this virus carrying this piece of DNA, that they would infect the same cells, that these two pieces of DNA would then get released into the nucleus of those cells, and they could find each other. And DNA has this capacity to recombine. And it's called homologous recombination. That's a technical term, but it basically means that you're taking two parts of a gene that have some overlap and you're recombining it into a full-length gene. So like I said, there is precedence for this. It has been done with other genes, but there's a little bit of luck involved to know if it can happen. So we made this system. This is approach number one. Approach number two, and this sort of goes in the order of complexity. This is the easiest thing to try, and then this is a little bit more difficult, and this is more complicated to design. So we made this, and then we also made this. This is a sort of a different approach, but the concept is the same. We still have the first half of the gene, the second half of the gene. But in this case, we're not trying to rely on them to recombine at the DNA level like we are in figure A. In figure B, what we're trying to do is have these things line up side to side. And we know that these AAV viruses, they do this if you get more than one copy of an AAV genome inside a cell, they will stack up in line against each other. And so what we've done is we've put the first half of the gene in one virus, and then the second half of the gene in the other virus, and in between we've added something called an intron. And this is a molecular biology term, but an intron is essentially a region of, in this case, RNA that gets cleaved away. The cells recognize this as a sequence that's not part of the gene, and it cuts it out. And then it fuses those two copies together at the RNA level, not the DNA level. So we're going from DNA to RNA to protein here. So we made this as well. We created an AAV that has the first half of SBF1. We put this intron sequence at the end of it. And we made a second one that has the other half of this intron and then the other half of the gene. And what we hoped is that these two viruses will line up together. They will create a long RNA that then, which is shown here, this sort of wiggly molecule, it represents an RNA. And then this piece, the gray and the gray, black, and gray here gets chopped out. And then the blue pieces, the half of the genes at the RNA level get cleaved back together or spliced back together. And then you can produce a full-length SBF1 gene at the RNA level. So that was strategy number two. And we made this as well. Okay, so the unfortunate thing is we've done this. So we made these, I guess, this summer. And we made the viruses. We injected, so we made 4 viruses. We packaged those into an AAV backbone, or an AAV, it's called the serotype, that likes to infect neurons. It's called AAV9. And we delivered these different systems, the A and B systems, in combination to mice. And so we did injections into baby mice, newborn mice, through their venous system. So we injected it into their blood. And by doing that, these viruses, they can travel through the blood and they can infect neurons. And then they obviously get into cells that way. And then we hope that we get this recombination. We did it that way. And we also did it by injecting muscle. Muscle's and we like muscle because it's easy to hit. Like in a mouse, I mean, muscle is, in human beings, muscle is 40% of our body mass. It's a big target. So it's really easy to inject viruses into muscle. So we figured we'd hedge our bets and try both ways. And unfortunately, we've done this experiment twice now. We haven't seen any evidence of recombination using this approach or this approach. It was disappointing, but probably not unexpected. These are they're not, and we know they're relatively inefficient. These were kind of a gamble to try these, but we thought it was worth trying. We haven't seen any benefit in it, and in fact, we did see in the group that was injected in the muscle, we had some mice that were injected with only this half of the SBF1 gene, and they actually showed a little bit of evidence of toxicity. I've got a question I can get to in a second. So, so bottom line is that we tried A and B, they didn't work. And now we're pivoting to, and so we have 8 months left on this project. And so now we're designing what we think is actually the most efficient and the most promising approach. It's called protein transplicing using inteins. And I'll tell you about this just a little bit. This is the reason we didn't begin with this, is because it's the most challenging one to design. And so we thought we'd try the easier ones to design first. They didn't work. So now we're pivoting to this. And so I mentioned that this works at the DNA level. This works at the RNA level. Now this works at the protein level. So what basically, it's the same concept. We're producing half the gene in both cases. But what we're also doing, this orange box is supposed to represent something called an intien. And these intiens are little pieces of protein that come from bacteria, actually. Bacteria have these mechanisms that they can naturally cleave or splice proteins together. So you can take one small protein and another small protein, and if they share these intien sequences, they can naturally paste themselves together. So this system has been shown to work for several different genes. It's like I said, it's a little trickier to design. And so this is what we're doing now. I'm showing here just, one, but we're actually going to try to make as many of these as possible. I think we're going to shoot for about 10 or 12 different designs, see which one works the best. I'm pretty confident that we're going to get one of these to work. And then once we have our lead sequence, we'll make those into these viruses and try to inject them into mice. And I think we can probably do that, by the end of the first quarter of 2023, we should be there. The cool thing about this is that this is, like I said, this is a natural system that comes from bacteria. So we know how it functions. And the bacterial sequences, once this protein splicing happens, bacterial proteins go away. So what you're left with is this fully recombined protein at the end. So that's where we are. I wish.
Not hearing Scott.
Yeah, Scott, I think you muted yourself.
Oh, okay.
It's
Sorry about that.
okay.
Yeah, so it must have happened when I stopped sharing.
Yeah.
So, I mean, this is science. It's the unfortunate thing is that 95% of our ideas don't work. It can be a very frustrating thing, frustrating field to work on. But this is why we decided to take this multiple step approach, because we wanted to give ourselves the best chance of having something work. We tried the easy things. They didn't work. Now we're moving on to the more complicated designs. And, I'm confident that we're going to get one of these working. So it's just a matter of doing the work and getting it done. And I don't know if you heard, if maybe I was muted, but I think we can have these ready, our new set of viruses ready to inject in mice probably by the end of first quarter 2023. So that's where we are. Like I said, I wish we had better news right now.
But you know what, Scott, we know we're unlucky, but we work really hard, and we're so happy to have you on our team who's working really hard and designing all these other things. And everyone knows me and Robin don't stop, and that's why we're so lucky to have connected with you, who doesn't stop. And we knew there were going to be hurdles along the way, and we're just so happy that we have you who's thinking outside the box, and we're ready to try the next step. So thank you for being such a great part. And I think that I didn't realize that Laura was on the call, and she is a researcher and she does have the question, and she's the one who has designed our rat model. So maybe you can answer her question because I don't even understand what it means.
Yeah, the question was about CRISPR. So so maybe you've heard about CRISPR. It just won the Nobel Prize recently, and the idea of CRISPR is to use these bacterial enzymes to you can design these to very specifically cut DNA and try to have recombination occur. You can try to repair mutations with CRISPR. It works really well in cells on a dish. And it actually works really well in what we think of as ex vivo gene therapy, where you remove a cell from a person, you can modify that cell in the lab, and then you can characterize that cell in the lab, grow it up in multiple copies, and then add the cells back. For something like a disease that affects neurons, that's more challenging, number one. But the other thing is that the CRISPR enzymes are bacterial derived, and we know there are immune responses to them. I don't think we have a good handle on the specificity. I think they can cut in more than one place, and we don't want to, inject this enzyme in somebody that's going to be there essentially permanently, that can potentially cut other places and cut up the genome and cause other problems. There's a paper recently that was published just last year from a lab at the University of Missouri, where they were trying to treat Duchenne muscular dystrophy with the same AAV vectors we're using, delivering a Cas9 enzyme, which is part of the CRISPR system. And they showed a massive immune response that was, it ended up being fatal to some. This was in dogs, to some dogs. And we're getting ready to publish a paper where we're using CRISPR enzymes also in vivo, which means we're delivering them to organisms with these viruses. And we're seeing evidence of what's called the cytotoxic T cell response, which is essentially a vigorous immune response that attacks the cells that have been infected by these viruses. So I think CRISPR is very powerful. I think the future is really bright for the technology. I'm not comfortable using it in a human being. I think there are many problems we still have to work out before we're there. And I can't give you a timetable on when CRISPR is going to be ready for in vivo gene therapy, but I don't think it's in the next several years. That's my opinion based on what I've seen and what's out in the literature. So I think it's an interesting question, but I'm not comfortable using it.
Well, Scott, thank you so much for taking your time and speaking with us tonight. We really appreciate you being here and being on Team Hunter, and we are excited to be moving forward to the next phase of the research, and words cannot express how lucky we are that we found you. So thank you.
Well, I appreciate that. You should hold those thankful words until we have our next round of data.
Well, you know what, Scott, at least you're working on the problem, because, when we started this, there was no research being done on SBF1 and no one trying to develop a treatment. And we know that all these treatments that are developed, there were roadblocks and hurdles along the way, and that you have to start somewhere. And so we're just happy to be getting everything together and starting and moving forward.
Well, thank you. I appreciate that. And if anybody has any questions, I mean, I know it can be complicated and there's a lot of scientific jargon. So if I confuse you all, please let me know. I'm happy to try to clarify anything. Do I see Ali? Did you raise your hand? Ali?
We can't hear you, Ollie.
Ali, you can't hear you.
I don't know. He's not muted. You want to type your question in the chat, Ollie?
There might be a button, Ali, on the side that says turn your audio on Wi-Fi or something. on your iPad. Or
Or just
whatever you're
push
gonna
your
use.
volume, push your volume button.
It's not the volume on, it's
It's
turning
the microphone.
the microphone on. It's on the Zoom app.
Or you could just type it in the chat.
Good typing.
Oh, it's a
We
typo.
hear you.
Can you hear me now?
Yes,
Yeah,
you're
okay.
staticky, but we hear you.
All right. The question that I have is that you said the first quarter of 2023, you will get, a successful, procedure, to go more forward. The question is that, is there any boundaries or limitation you think of, or you're good to go? Just the time is important for us.
Ali, we're having trouble hearing the question.
Yeah, you're still we hear you, but you're staticky.
Ali, maybe you can type in the question. But meanwhile, Scott, I have to thank you for taking part in this and helping us. We're very, very excited, and Iris and I remain very, very hopeful to find some kind of treatment for these kids. So we're very excited.
Thanks, Robin. I appreciate that.
Well, Iris, maybe we should move on, and then Ali can always come back and he can come back to this.
He's
Yeah, I mean, if you want to, actually, I can leave my email in the
Yeah.
chat.
Yes,
And
that would
that
be
way,
great.
If you want to just send me an email, I'm happy to answer by email as well.
That's perfect. And we can also share your email if you're okay with that with him.
Sure, yeah.
So then I am going to go back to sharing my screen and promise we will wrap this up soon, guys. Okay. Brett, are you going to speak?
First of all, I wanted to thank all of my friends and family for coming tonight and helping us raise funds to save my son Hunter and other kids and young adults with CMT4B3. 100% of the money raised goes directly to fund research projects. And I also want to give a big thank you to our executive board, Leanne, Jared, Mandeep, Amy Schwartz, Lindsay Zakin, for their advice, wisdom, direction, and support to help us achieve our ambitious goals of finding a treatment to stop the progression of this debilitating disease.
Thank you. And then why we're here today. So now onto the part where we need your help. Giving Tuesday is less than 2 weeks away. It's November 29th. It's the Tuesday after Thanksgiving. End of year giving is where foundations make the majority of their money. Giving Tuesday is the start of giving season. So we are excited to share that we are super close to our initial $1 million goal. We have raised $965,315 so far. And we have some very exciting news to share. A donor has come forward and has pledged $50,000 and is going to be matching every dollar raised up to $50,000 for Giving Tuesday. This means that every donation has double the impact. We learned from last year that you guys sharing our story with your friends and your family, your network makes such a big difference. It helped us raised, I think Giving Tuesday alone, we raised almost $100,000. And since we started the peer-to-peer fundraising campaign, we raised over $350,000. And that's all because of our friends and family. And we wanted to make it as easy as possible this year for you guys to help us meet our goal, which we know we can make because we have this match. So one of the easiest things you can do is to create a Facebook fundraiser. I'll be following up with an email, and all you have to do is click the link. Everything is pretty much pre-automated, and then you just invite your friends. It's really easy. Facebook is also doing this cool thing this year where they're matching reoccurring donations. So anybody that's a reoccurring donation, Facebook won't match that up to $100. The second thing you can do that's really easy is to select us on Amazon Smile. It's also a link that you just click and it sets it up for you. I will send that as well. If you do this, Amazon will then donate 0.5% of every purchase you make on Amazon. It doesn't sound like a lot, but it definitely adds up. Each quarter we get $400 or $500, and it definitely, every little bit makes such a big difference. And the more people we can get to sign up, the more money we will get. And the third and most important thing that we are asking of you is to personally reach out to your network and share our story with your friends and family. And last year we had the peer-to-peer campaign, but we found that, we were walking so many people through how to do it that we wanted to make it a little easier for you. So we drafted an email template that you can personalize, and you can email it to your friends and family. So I have this, and I also added a picture of Hunter because everyone likes a picture of Hunter. So this is what the email says, and it says, "Dear family and friends, as you may know, next Tuesday is Giving Tuesday. I hope you will join me in supporting my dear friends Iris and Brett Schultz as they race to develop a treatment for their 5-year-old son, Hunter. When Hunter turned 3, he was diagnosed with Charcot-Marie-Tooth disease type 4B3, a life-threatening, ultra-rare progressive neuromuscular disease." You know what? I realized I think I might have put the wrong template in here. Nevermore. It's fine. It's along these lines, and I will send it to you later.
That's
And
not the correct one, but the
that's
correct
not the
one.
correct
both
one,
times.
but it's something similar to this, and it's going to have a link to donate, and it's going to have our website and the video you can watch, and it's basically something like this, and that is pretty much it. So we really hope that you guys will help us this giving season to reach out to your family and friends and to share this email. You can change it however you see fit. But that is it. Oh, we also, if I can get this to work, we're going to share with you a video. I see the chat has some stuff going on here. Okay, so share this video that University of Miami put together for us for Rare Disease Day, just to show you how stop share. Sorry, I was having technical difficulties here before, guys, getting my volume working. So I'm just glad we made it through the presentation. Bear with me 1 second. Okay. Let's see if this works. You guys tell me.
Well, while we're waiting for Iris, I want to show everyone, this is the new issue of, I don't even know if Scott knows about this, University of Miami Medicine Magazine. It comes out twice a year. And it's about basically medicine's new frontiers. And then in the centerfold, who could that be? It's Hunter and Iris, and there's a whole article about DNA detectives, a family's journey to cure rare genetic disorder. There's a whole article about us. So also we're getting a lot of press for the Charcot-Marie-Tooth community, and that's really wonderful.
That's great.
Yes, yes. So a lot of scientists are taking interest in us because it is a peripheral nerve disease, and it's wonderful.
Let's see if we can share the video that they played. Do you see it?
Yes.
Do you hear it?
We see it. We don't hear it yet.
It's not playing
It's
yet.
it's not on. You gotta click that arrow.
No,
There we
it
go.
was playing on my computer, but the problem is for some reason YouTube is not being very friendly today. Do you see it now? Hear
Yes.
it?
Yeah, we're good.
Hunter is 4 years old. He was a healthy, happy little boy, and he just lit up our world. We started to notice something was different with Hunter when we would take him to little baby classes. He was always behind. They would be rolling over. Hunter wasn't rolling. They were sitting. Hunter wasn't sitting. As a mom, something didn't feel right. It took us 3 years to get the diagnosis. CMT4B3 is similar to muscular dystrophy, but instead of the muscles wasting away, the nerves deteriorate, which causes muscle weakness and eventually muscle atrophy, and patients lose the ability to walk, to talk, the use of their hands.
A rare disease is generally defined as less than 200,000 patients in the United States. So what we do for Hunter here in this lab is really a very advanced idea. We want to bioengineer the protein that he's missing.
Dr. Stefan Zuckner is trying to develop a mini gene for us. We're rewiring Hunter. We're permanently changing his genetic makeup. We're getting rid of the faulty genes and replacing it with a functioning copy.
So genetic therapies are on the rise. One very powerful technique is to really replace a faulty gene with the correct copy, so that hopefully he can live a fairly normal life.
He's taken CMT4B3 on, and he wants to solve this problem with us.
Just because something is ultra rare doesn't mean there is no hope. With new technology now, we can actually identify the genetics underlying these ultra rare diseases.
We're really lucky that Hunter is part of the family now, and that we get to have access to all these amazing, brilliant researchers and doctors and scientists that, wouldn't even be thinking about CMT4B3 if it wasn't for him. It's really paving the way for genetic treatments. If this works, we're potentially stopping the disease in its tracks.
So
Pretty amazing.
that was, we're actually funding 2 different research projects at University of Miami, one with Stefan Zuckner, who's doing the gene therapy, and then with Mario Saporta, who's taking Hunter stem cells and other CMT4B3 patients. Uh-oh. Sorry. Various, and Mario Saporta, who's taking these stem cells from Hunter and other patients and examining them to see if we can test already FDA approved drugs on them, to see if we can potentially find a treatment that's a drug that's already approved that we could start giving our patients right away. So we're just really happy to be working with so many researchers who are collaborating together and know that time is of the essence and helping us find a cure.
Great job.
I know Robin wanted to say something. Robin.
Okay, I just want to say a few things that actually I wrote down. First, I want to thank everyone for coming. It's so wonderful and supportive for us emotionally. It's just great to see everybody. And Scott Harper, thank you so much for being here. I wanted, Iris gave you the update on where all your research dollars are going, and it's very, very exciting. And as you know, 2.5 years ago, my grandson Hunter was diagnosed with a rare disease, with Charcot tooth disease. And of course, we had never heard of this. And we were told that it was a degenerative neuromuscular disease. And we were also told it was caused by a mutation in one little gene, as Scott told us, SBF1, but a very important gene that keeps the nerves, the coating around the nerves healthy. So we have to fix this. Hunter has difficulty with his vision, walking, and his speech, yet he's smart, inquisitive, active, and a happy little 5-year-old. So just a few statistics. There are over 7,000 rare diseases affecting over 300,000 people worldwide, and half of those affected are kids, just like Hunter. Of these kids, 30%, that means 3 out of 10, don't live to see their 5th birthday. So we're lucky. Hunter is now 5. And of these 7,000 rare diseases, only 5% of them have a cure. So we quickly learned we had quite the challenge ahead of us. Hunter's diagnosis has been bittersweet. Yes, after 3 long years of seeing every specialist in the tri-state area, we finally had an answer. But there was no treatment, no cure, and no research being done for this rare genetic disease. So Iris and Brett and our family stepped forward to be the first, and we met fabulous scientists like Scott who could help us. So now these statistics. 3 million people worldwide have CMT, and no one's heard of this disease. It's absolutely crazy. No treatment, no cure for any form of CMT. CMT usually starts in the 4th or 5th decade of one's life. So how many kids have Hunter's early onset, ultra rare type of CMT4B3? Good question. We don't know. Many of these kids have been misdiagnosed with muscular dystrophy as young adults, ALS or CP. So Iris and I are changing the platform for diagnosis and understanding of this disease in the scientific community. So this goes way beyond Hunter. Hunter's infectious laugh and determination inspires me each day to keep fighting for him and all these other kids. I want to see Hunter run, walk, jump, and continue to be able to play with the other kids on the playground. But without a gene therapy, this will not happen. So we have an amazing team of scientists that we have put together, and they collaborate and work together. It's amazing. And the only thing that's standing in our way is raising money for the research. Only 5 years ago, a treatment would not have been possible. But today, the science that can create a treatment for kids like Hunter is totally possible, and gene therapies are being administered all the time. However, we are realistic, and we know that we may not find a treatment in time for Hunter to maintain his mobility, but we will find a treatment for children that come after him and the children that come after them. And that wakes me up super excited every morning as I fight to save the lives of kids with neuromuscular diseases. So I just want to thank all of you for participating and letting you know you're truly, when you donate, making a big difference and really moving science forward. So our family is so greatly appreciative to have all of you, friends, family, just donors, people that came tonight that I'm not even sure who they are. I'm looking like, who is this? So I'm really excited. Thank you all so much.
Oh, and somebody else wanted to say thank you.
Thank you everybody for coming to the essay.
Okay, I'm going to stop